Ever notice how, like, a cut on your finger just kind of heals itself?
Yeah,
It's pretty amazing when you think about it.
it is.
Well, our intestines are kind of like that, too. I mean, they're
Right.
constantly repairing themselves from all the stuff we put them through.
Right, all that wear and tear every single day.
Exactly. It's kind of mind-blowing.
It really is. And it's something most people probably don't even think about.
Nope. We just kind of take it for granted.
Yep, until something goes wrong upon it.
Right. Which is exactly why this new research is so fascinating.
Oh, absolutely.
It's coming out of the University of Colorado Anschutz Medical Campus,
Uh-huh.
and they're really digging deep into how our intestinal cells manage to regenerate like that.
It's really groundbreaking stuff.
It is, and we're taking a deep dive into a Science Daily article about it,
Okay.
which is based on a paper that was just published in Nature Cell Biology.
Very cool. So for this deep dive, we're going to try to really break down this research.
Yeah.
Understand how these intestinal cells renew themselves
hmm
and what it could mean for treating diseases like cancer.
Exactly. And we're going to do it without getting bogged down in all the technical jargon.
Right. Make it accessible for everyone.
Exactly.
Sounds good.
So let's start with the basics. The researchers at CU Anschutz were trying to figure out what triggers this whole regeneration process.
Right. Like, what's the signal that tells a regular intestinal cell to suddenly become more like a stem cell when there's an injury?
Yeah, it's like they're hitting a reset button or something.
That's a great way to put it.
Yeah.
I mean, think about it. These mature cells have very specific jobs, right?
Uh-huh.
Like absorbing nutrients or protecting the intestinal lining.
Right.
But when there's damage, they have to switch gears and become more versatile
Okay.
so they can divide and repair the injured area.
And the researchers wanted to understand exactly how that happens.
Exactly.
Yeah.
Like, how does the cell know when to make that switch?
Yeah, what's the signal?
Right.
Well, the guys who cracked this case are Peter Dempsey.
Okay.
He's a professor in pediatrics developmental biology. Uh-huh. And Justin Brumbaugh, an assistant professor of molecular, cellular, and developmental biology.
Gotcha.
And using animal models, they zeroed in on this process called H3K36 methylation.
Okay, now that sounds pretty complicated.
It does, but it's actually a pretty elegant mechanism when you break it down.
Well, let's break it down then. What is H3K36 methylation?
So basically, it's a tiny chemical modification that affects this protein called H3 histone.
Go ahead.
And histones are these proteins that our DNA wraps around.
Interesting.
Like spools of thread, almost.
Okay, I get it. So these modifications to the histones can affect how the DNA is packaged.
Exactly. And that, in turn, determines which genes are active in the cell.
Oh, wow. So it's like a master control switch for gene expression.
Exactly. And what they found is that H3K36 methylation acts as this on-off switch for the regenerative state of intestinal cells.
Okay, so how does that work?
Well, when this methylation is present in a certain way, it keeps the cells doing their normal specialized jobs. But when there's an injury, the methylation pattern changes.
Mm-hmm.
And that allows the cells to revert back to that more flexible stem cell-like state.
So it's like flipping a switch.
Exactly.
Yeah.
And this tiny molecular tweak is what unlocks this amazing regenerative potential.
It's incredible how these tiny changes at the molecular level can have such a big impact.
I know it's mind-blowing, but it makes sense when you think about it.
Also.
Well, most of the time, these cells need to be focused on their specific tasks, right?
Okay.
Like, you don't want them randomly deciding to become stem cells all the time.
Yeah, that would cause chaos in the gut.
Exactly. So this H3K36 methylation is like a safeguard.
Okay,
It prevents this unwanted flipping of cell identity.
that makes sense.
And you know what's really interesting?
Yeah.
This uncontrolled switching back to a less specialized state is actually a hallmark of cancer cells.
Oh, wow. So this research could have implications for cancer treatment as well.
Absolutely. But first they had to prove that this methylation process was actually happening in living organisms.
Right, because you can't just assume that what happens in a petri dish will happen in a living animal.
Exactly. So they used animal models to actually observe this whole process in action.
And what did they find?
Well, they were able to see that changes in H3K36 methylation directly correlated with the cell's ability to regenerate after injury.
Wow, so they actually saw it happening.
They did, and that was the key piece of evidence linking this specific biochemical process to what they call cellular plasticity.
All right, and cellular plasticity basically means the cell's ability to change its state or identity.
Right. In this case, from a specialized intestinal cell back to a more stem cell-like cell.
Gotcha.
So they've identified H3K36 methylation as this master regulator. Now the question is, what can we do with that knowledge?
Where do we go from here?
Well, the researchers are already thinking about how they can target this process for medical treatments.
Like how?
Well, their main focus is on colorectal cancer and precancerous conditions like chronic colitis and inflammatory bowel disease.
Okay, so how does this methylation process play into those conditions?
Well, we know that chronic injury, like what happens in inflammatory bowel disease, can increase the risk of developing colorectal cancer.
Right, that makes sense.
So the idea is that if we can understand and maybe even manipulate this regenerative switch, we might be able to intervene in these diseases.
So potentially preventing those precancerous conditions from developing into full-blown cancer.
Exactly. Or even promoting healthy regeneration in damaged tissue.
Wow, that would be huge.
It would. And the potential applications go beyond colon cancer.
Really?
Yeah, the research suggests that this methylation process might also play a role in why some cancer cells become resistant to chemo and radiation.
Oh, that's a big problem in cancer treatment.
It is, and if we can figure out how to control this switch, it could lead to more effective treatments.
I see. So by either preventing that resistance from developing or making the cancer cells more vulnerable to existing treatments.
Exactly. And they're also looking at whether we could protect healthy intestinal stem cells during cancer therapies.
That's interesting because chemo and radiation can really damage the intestines, right?
Right, but if we could temporarily activate this regenerative switch in healthy stem cells, it might make them more resistant to those treatments.
And that could potentially reduce the side effects for patients.
Exactly.
This is all pretty amazing stuff, but it sounds like it's still early days.
It is. But identifying H3K36 methylation as this key regulator is a really important step.
Yeah, it gives researchers a specific target to focus on.
Exactly. And Justin Brumbaugh himself said this research is just the beginning. It's a foundation for some really exciting possibilities down the line.
Like what?
Well, they're talking about using this knowledge to develop better ways to test new drugs.
Okay.
And to create more realistic models of diseases in the lab.
That would be helpful.
It would. And maybe even someday creating specific cell types for transplantation therapies.
Wow, so we could potentially guide cells to regenerate in a very targeted way?
Exactly.
That would be incredible.
It would. And it all comes back to this fundamental research into how our cells work.
Right. By understanding these basic mechanisms, we can unlock new ways to treat all sorts of diseases.
Absolutely. And just to be totally transparent,
Yeah.
this whole deep dive has been based solely on the information released by the University of Colorado Anschutz medical campus about their paper.
Right, we haven't gone beyond that report.
Nope. Just trying to give a clear and understandable overview of this research.
And its potential implications.
Right. So let's recap. The big takeaway here is the discovery of H3K36 methylation
Okay.
as this crucial regulator of intestinal cell regeneration.
Mm-hmm.
It's like this on-off switch that determines whether cells can revert to a stem cell-like state after injury.
Right.
And this finding opens up all these new possibilities for treating colorectal cancer and other intestinal diseases.
By giving researchers a specific target to manipulate.
Exactly. And this leads us to a final thought for our listeners. If we can actually control this cellular reset button in the intestines, what other regenerative powers might be hidden within our bodies?
That's a great question. Like, what other possibilities are out there just waiting to be discovered?
Exactly.
Yeah.
And what are the implications of being able to manipulate these fundamental biological processes?
Yeah. It really makes you think about the future of medicine
It does.
and the potential for healing that exists within us all.
Really fascinating stuff.
It is.